Hi everyone, good day. This is Dr. Von, and for today, we will be discussing about your principles of automation in your hematology laboratory. This will be your third part of your laboratory evaluation of cells. You already discussed the different manual laboratory evaluation of cells, but right now, ang i-discuss naman natin is all about the automation, and most specifically the principle behind this automation. Kasi majority of our laboratory right now are using automated machines. Pero there are still some na mga, what you call this one, primary laboratory that you still have to perform your manual testing. And also, even if that you are already in a you are using automated machine, if there's something wrong with your machine, we usually double check it through manual testing. And also, we have naman controls for your automation. Your automated blood cell analysis has virtually replaced your manual hemoglobin, hematocrit, and cell counting because of its greater accuracy and precision. We could release the result as soon as possible, and you can perform many tests in one run or in one series of testing. Your hematology blood cell analyzers are produced by multiple manufacturers, including but not limited to Abbott Laboratories, your Horiba, your Siemens Healthcare Diagnostics, and your Beckman and Coulter. One of the most common na mga machines that you will encounter is really your Beckman and Coulter. We also have your Sysmex Corporation. These are the four major brands or manufacturer of your different hematological analyzers. But before we go to our discussion for today, at the end of this video lecture, the students will be able to explain the different principles of your automated blood cell counting and analysis. Describe how the general principles are implemented on this different instrument, and we have to identify what are the parameters that can be measured on these analyzers. Then I want you to interpret your white blood cell, red blood cell, and platelet histograms, and then explain the general principles of your automated reticulocyte counting. And lastly, the most important part is to identify the different sources of error in automated cell counting and determine the appropriate corrective action. The man who started it all, it was your Wallace Coulter and Joseph R. Coulter. So the automating the hematology laboratory, ito yung tinatawag nila na the Coulter legacy. So Wallace Coulter was actually a very humble and a caring employer who treated the people at every level of the company as though they were members of his family. He actually never finished his college undergraduate program, yet he received numerous honorary doctorate degrees and served as an adjunct professor at the University of Miami in Florida. He held 74 patents and was received many scientific professional awards together with his brother, Joseph R. Coulter. They actually led the Coulter Corp, which they founded in 1958, to manufacture and distribute instruments that Wallace had invented based on this Coulter principle. So later on, that we will be discussing the importance of that Coulter principle. The most widely used method for counting and sizing microscopic particles suspended in a fluid. Actually, no, his method has been called the first viable basis for your flow cytometry, and from it grow an industry that forever changed the world of your diagnostic medical research. Again, the Coulter brothers were the early pioneers in the different automated hematological tests such as your cell counting, immunophenotyping, even the diagnosis of your hemoglobinopathies, diagnosis of your leukemias and lymphomas, and your coagulation abnormalities. There were also the early pioneers of your monoclonal antibodies, which have since proven to be extremely potent in helping the diagnosis of different types of cancer, such as your lymphoma, leukemia, and other diseases. So as a general overview about your automation, Doc, what are the advantages versus disadvantages of your automated machines? We all know, of course, number one advantage, speed and efficient handling. Most physically, if we are talking about large number of samples. Next, greater accuracy and precision. Most specifically, if we are talking about quantitative blood tests. Then, it is able to perform multiple tests on a single platform, and it is significant in reduction of your labor requirements because it is more efficient workload and management, more timely diagnosis. However, there are still disadvantages of your automation that you have to take note. Number one, flagging. Anong ibig sabihin ng flagging na yan? So, we have this term flags, meaning flagging of a laboratory test result demands your labor-intensive manual examination of your blood smear. Kasi for example, nag-run ka ng CBC, then it will create different flags if it will find something abnormal or that is not normal from that analysis. So what you'll do, of course, you will do now your manual examination. So kumbaga, automated ka na, nagmamanual ka pa because you have to double check. So that's the disadvantage, you're flagging. Comments on red cell morphology cannot be generated. Your RBC morphology cannot be generated. So therefore, abnormal red cells or red blood cell shapes such as your fragmented na mga cells cannot be recognized by your automated machine. And then, erroneously increase or decrease results due to interfering factors. So later on, we will be discussing what are those possible na mga interfering factors that will result into erroneous result. And ang pinaka disadvantage talaga niya guys is it's very expensive with high running cost. When I'm talking about expensive, literally we are talking about millions. Natatawa nga kami sa sobrang liit na mga different machine namin sa hematology, no? It will cost us 1 million, 2 million, 3 million. Usually sa mga laboratory guys, meron tayong mga medical representatives and usually they will be the one that will somehow endorse their machines. Tapos, however, since most of the laboratory kasi, we are not actually buying the machine. We are actually buying the reagents. Kumbaga, if we buy the reagents from that particular company, then they will allow us to borrow their machine. Pero, kailangan dun kami bibili ng reagents for that particular na machine. So, kumbaga, naka-tie up or may kontrata kami. So, kasi it's very expensive. The machine is very expensive. Millions. That's why usually naka-tie up or kontrata lang yan and bibili lang kami ng mga reagents sa kanila. So, pinapahiram lang kami na kanilang mga machines. So, these are the different types of your hematology analyzers. We have your semi-automated, measures only few parameters. Some steps like dilution is carried out manually. So semi-automated. So still, there are certain steps that you have or you need to do it manually. While your fully automated machine measures multiple parameters and it will require only anticoagulated blood samples. Let's talk about the basic components of your hematology analyzer. So we have your hydraulics, we have your pneumatics, and your electrical. So at least we have an overview of what are the different components of your hematological analyzer. If we just look at this one. It's very complicated, no? Like, oh my God, ano ba to? You know what, guys? If there's something wrong with the machine, of course, there are engineers naman for that particular machine or company, no? Usually, binibigyan lang kami ng contact number if there's error. Kasi usually, nag-error, maglalabas siya ng particular na warning na there's an error on this particular part sa display. So, we will just tell, for example, if we cannot really do it on our own, of course, we have to contact the engineers of their company. But still, we have to be aware or knowledgeable of the basic components of your hematological analyzer. May mga times nga, guys, na ang tagal talaga mag-reply ng engineer. So, ang ginagawa namin, gina-check namin ito, isa-isa, asan kaya? Parang kasi mag-flag siya na may clog sa particular na area. So, tinitingnan namin. May times kasi yung mga engineers, tinitingnan din namin kung anong ginagawa nila para at least if ever may ganun pa rin na error, at least alam na namin kung ano nang gagawin. So, but still, you have to contact the assigned personnel, no, to perform that one. Kasi remember, the machine is very expensive, millions. Makakasira ka, patay. Makakabayad ka ng million. So at least we are aware of the different hematological components of your analyzer. We have your hydraulics. Nandyan yung mga mixing chambers, yung aperture bath or your flow cell, and your hemoglobinometer that will read your hemoglobin result. Then, you have your aspirating unit, mga dispensers, and your diluters. So, nakakita nito, nandito na yung mga dispensers, mga diluters. Diyan lalabas yung mga different natin na reagents. And then, we have your pneumatics. Yung pneumatics, these are system that generates a vacuum and pressures that is required for operating the valves as well as open or moving the sample through your hydraulics system. Yung electricals naman, this control the operational sequences of the total system and includes your electronic analyzers and computing circuitry for processing the data generated. So, kumbaga, pinaprocess na dito yung ating mga data na nakukuha. So, again, remember these three basic component of your hematological analyzer: hydraulics, pneumatics, and your electricals. At least aware kayo of what is the function of these different components. So again, we will be talking about your principles of your automated blood analyzer. So ito yung very important principles or concepts that you have to understand. We have your electrical impedance, your light scatter, and your fluorescence. Sige, first, let's talk about your electrical impedance. So, ito yung sinasabi natin na we're talking about your Coulter principle. So, when we say Coulter principle, basic description lang muna tayo. Detection and measurement of changes in your electrical resistance produced by cells as they traverse a small aperture. Again, ulit-ulitin natin. You detect and you measure changes in your electrical resistance. So again, when you hear the word Coulter principle, think of electrical resistance produced by the cell as they traverse a small aperture. So that's your Coulter principle. So let's take a look at this diagram. So as what you can see, meron tayong tinatawag na aperture bath. Itong pinakamalaki. This is your aperture bath. Tapos, meron tayong aperture current, yung external electrode, and your internal electrode. And then, Doc, ano yung parang fluid na yan dyan? This is actually your blood cell suspension. Ano yung solution na yan, Doc? It's a composed of electrically conductive diluent. Again, this is your blood cell suspension, yung parang fluid dyan. It is a solution that is composed of electrically conductive diluent. Tapos, meron tayong parang tube dito. This is what we call now your aperture tube. Tapos, may butas sa ating aperture tube. Yan yung tinatawag natin na aperture. So, again, review natin. May dalawang chambers filled with a conductive buffered electronic solution separated by your glass tube having a small aperture. So there is a direct current is generated between your there's a direct current here between your external and your internal electrode. Again, let's review ha. Ulitin para maintindihan. This is your aperture bath. Merong aperture tube. Tapos may butas yung aperture tube. Tinatawag natin na aperture. Tapos may dalawang current tayo. External electrode and your internal electrode. And there is also a presence of your fluid. Ang fluid na yon is composed of electrically conductive diluent. I hope that is clear. So, anong nangyayari, Doc Von? Paano yun nangyayari yung Coulter principle? Again, Coulter principle, detection, measurement of changes in your electrical resistance produced by the cell as they traverse through a small aperture. So ano nga, saan nga ang small aperture? As they traverse your small aperture, dito. So yung cells natin magproduce ng electrical resistance pag dumaan sila sa ating aperture. So ano bang nangyayari? Again, your cells is suspended in a electrically conductive diluent such as your saline are pulled through an aperture or your orifice in a glass tube. In the counting chamber or transducer assembly, your low frequency electrical current is applied. So there is an application of your electrical current between now your external electrode and your internal electrode. External electrode suspended in the cell dilution, internal electrode housed in the aperture tube. Then, electrical resistance between the two electrodes or impedance in the current occurs as the cell passes through the sensing aperture, causing now voltage pulses that are measurable. I hope that is clear, no? As your cells traverses this part, again, as the cells traverse this part, electrical resistance between your two electrodes or your impedance current occurs as a cell passes through this sensing aperture, causing now a voltage pulses that are measurable. So again, balik tayo sa principle. You detect and you measure the electrical resistance na ating nagawa kanina by the cells passing or traversing your small aperture. As simple as that. That is your Coulter principle. I hope that is clear. Then, but you have to take note that your aperture for your RBC or platelet is smaller than your WBC aperture. So, iba-iba sila ng sizes kung anong cells ang ating dinetect. Of course, malaki ang WBC, therefore, malaki ang WBC aperture. Pero, since maliit yung RBC and platelet, of course, mas maliit yung aperture dito ng ating RBC and your platelet. Again, di ba, mag-generate siya ng voltage pulses that are measurable. Kindly take note that one pulse is equivalent to one cell. Again, one pulse is equivalent to one cell. So that's your Coulter principle. So after that, after generating the volts that you were able now to measure the changes of your electrical resistance, then some devices or some instrument may have a display of what we term as your oscilloscope. Ano yung oscilloscope, Doc? Your oscilloscope screens on some instrument display the pulses. Now, remember, it will display the pulses that are generated by the cells as they interrupt the current. The number of pulses, ang number ng pulses na ito, is proportional to the number of cells counted. So, kung ilang pulses dyan, that would mean the number of your cells that you're able to identify. However, the height, so ang height nito, from here, here, the height of the voltage pulse is directly naman proportional to the volume of the cell. which allows the discrimination and counting of cells of specific volumes through the use of your threshold circuit. So again, sa oscilloscope, anong ibig sabihin nito? Bawat pulse na dyan, that would mean the number of cells counted. Yung height naman dyan, that would mean the, ano nga height? Remember, that is proportional to the volume of your cell. Kaya nga nakalagay dito, femtoliter. For example, this one cell, anong volume niya? So, tingnan natin, itong cell, ito pinakauna. So, nanda na around 92, below 92. For example, ito, pinaka-last na pulse natin, around between 92 and 90 femtoliter ang kanyang volume. So, again, pulses are also collected and sorted according to their amplitude by pulse height analyzers. Then, after that, the data are plotted on a frequency distribution graph or your volume distribution histogram. Meaning, you could also present your data as a histogram with relative number of the y-axis. Anong y-axis dito? This is your y-axis. Relative number, we're talking about the number of your cells versus the x-axis. Again, talking about the channel number equivalent to the specific volume or volume of your volume on the x-axis, which is your femtoliter. The histogram produced depicts the volume distribution of the cells counted. So later, sige lang, we will have a separate discussion of your histogram. So again, from your Coulter principle, you can present that as in a oscilloscope screen or through a histogram. Kumbaga, ang diagram na to from our principle na na-discuss kanina, you can now present that as an oscilloscope screen sa mga old display natin, or could be in a histogram distribution. Don't worry, we will discuss your histogram later. Okay. However, there are several factors that may affect the volume measurement in your impedance or volume displacement instrument. Number one of the most important factors that we have to take note is the protein buildup. So again, sige nga, pag may protein buildup dito, ano kayang mangyayari sa ating cell count? Will that be result into increase or decreased cell count? Say for example, meron tayong buildup ng ating proteins dito. Would you think that will it result into an increased cell count or decreased cell count? Sige na, I want you to answer on your own. Hindi ko naman maririnig kung ang sagot niya. Sige na, just try. Increase or decrease? Of course, it will result into lower cell count kasi nga magbi-build up. So, do you think that the cell can still pass through to that small aperture? Of course, mahihirapan ang cell. So how about the cell volume? Will it cause an increase or decrease of your cell volume? Of course, what if may build up dito, malaki, dumadaan yung mga malalaking proteins, magbi-build up, of course, nabablock yung cell count. How about the cell volume? Will it cause now an increase or decrease? It will create a falsely elevated cell volume because remember, your proteins are bigger. However, there are current instruments that incorporate your burn circuits or other internal cleansing system to prevent now your slow protein buildup. Again, may mga machines na nagdadagdag sila ng burn circuits para nililinisan yung cleaning system para ma-prevent or ma-slow down yung protein buildup. We also use your hydrodynamic focusing. Ano yung hydrodynamic focusing? The use of your hydrodynamic focusing avoids many of the potential problems, including your protein buildup. Bakit? Ano yung hydrodynamic focusing, Doc Von? It provides a sample stream surrounded by a sheath fluid as cell pass through the aperture. So, for example, ito yung aperture natin dito na area. Diba? This is the aperture. This is now the aperture. These are the cells. As what you can see, it is surrounded by a sheath fluid. So, ito is a fluid ito. It's surrounded by a sheath fluid. What will happen because of that sheath fluid? It will now prevent coincident passage of cells and recirculation. This is recirculation, I'm sorry. Recirculation of your cells. Again, laminar flow allows the central sample stream, ito yung central sample stream, to narrow sufficiently to separate and align the cells into a single file. So, at din, nakikita natin na nag-a-align yung ating cells into a single file for passage to the sensing zone. Then, the outer sheath fluid. Itong outer sheath fluid ito na nakapalibot sa ating aperture para maging align yung ating mga cells. Anong nangyayari? The outer sheath fluid minimizes your protein buildup and plugs, and it will eliminate again recirculation of cells back into the sensing zone. So I guess we were able to address the problem of a possible protein buildup through your hydrodynamic focusing because, diba, may sheath fluid tayo na naka-surround. I hope that's clear. So these are the different variables measured by your electrical impedance. Meron tayong parameter sa ating red blood cell, parameter sa ating WBC, parameter sa ating platelet. So ito yung tinatawag natin ng mga parameter sa RBC, red blood cell count, your MCV, your size distribution histogram. Don't worry, we will discuss histogram later again. Your red cell distribution width, your hematocrit, your MCH, and your MCHC. You have also your WBC parameter, your total count, your three-part differential count, your lymphocyte, monocyte, and your granulocyte, your BEN, your basophil, eosinophil, and your neutrophil, and your platelet count. Your platelets, including your platelet count, the histogram, the platelet volume, and platelet distribution width. So usually kasi pag histogram, we are talking about the distribution of a particular cells and its volume. Makikita natin yan later. Now, we're done with your electrical impedance. Now, this is the another concept that I want you to understand. Your radio frequency. May mga machines na hindi lang gumagamit ng electrical impedance, but also it uses your radio frequency. Kumbaga, it's a combination of different principle. So, may impedance na, hahaluan mo pa ng radio frequency. So, again, this is again, di ba, we discuss your low voltage direct current impedance or your electrical impedance as described a while ago that can be used now in conjunction with your radio frequency resistance. So again, dalawang factors. Ihahalo natin yung electrical impedance plus RF frequency. So two parameters ang i-discuss dito. Yung radio frequency or your direct current, yan yung tinatawag natin na RF DC detection method. Kasi dalawang method, impedance and radio frequency. Your RF DC detection method. It will show simultaneous use of your direct current and your radio frequency in one measurement system. Conductivity as measured by your high frequency, yung mini-measure ng ating radio frequency which is your conductivity, is attenuated by your nucleus to cytoplasm ratio, nuclear density, and your cytoplasmic granulation. Again, yung conductivity na mini-measure that is measured by your high-frequency electromagnetic probe is attenuated by your nucleus cytoplasm ratio, nuclear density, and your cytoplasmic granulation. And there will be two different cell properties because we are using dalawa ang ating ginagamit, direct current and radio frequency. So therefore, we will be able to produce two different cell properties such as your low voltage DC impedance and RF resistance that can be plotted against each other to create now your two-dimensional distribution cytogram or your scatter plot. Such plot display now the population of your cluster, the cell population as clusters, with the number of dots in each cluster representing now the concentration of the cell type. Again, Doc, napaka-complicated naman yung explanation natin about radio frequency. Ganito lang. Gusto ko lang isipin nyo, yung radio frequency, in-add natin sa diniscuss natin kanina yung electrical impedance. Diba? We all know, alam na natin yung electrical impedance kanina dito. But in radio frequency, it can be added to your electrical impedance. With that being said, dalawa na property ang ating makikita and mapaplot sa ating display. Makikita na natin yung cell distribution as clusters and what else? Yung number of dots in each clusters, yung mga dots na dyan will represent the concentration of your cell type and as well as the clusters of cells. So, number and cluster, or pag sinabi natin clusters, that's a particular group of cells. So, naka-cluster dito, monocyte, naka-cluster dito, lymphocyte, that's a cluster. So, that's another important parameter or property na nakukuha natin. Clustering and the dots representing the number of cells. That's it. As simple as that. Gusto ko, yan lang ang ating thinking. Do not overthink na lang, okay? I know it's very complicated, no? It's so physics. Me too, I don't like physics. I guess, no, ang pinaka-importante naman dito is that you really understand, just understand the very simple concept, because at the end of the day, no matter how complicated the question, as long as you understand the concept, then you can still answer the question. Now, another important concept, that's your optical light scatter. Sige, tingnan natin your optical light scatter. So, dito yung dumadaan ang ating cells. So, tingnan nyo, these are the different WBC cells. So, tingnan na, do not read ha, huwag nyo munang basahin. Gusto ko muna ipakita sa inyo yung parts ng ating optical light scatter. Meron tayong flow cell, dito dadaan, or laminar flow, which is a single file, dito, isa-isa ang ating cells. Meron tayong WBC, ito yung mga WBC natin. Meron tayong lasers sa side, your laser that will emit a monochromatic light. Again, your laser will emit a monochromatic light. Ano yung different light sources natin doon? We have your tungsten halogen lamp or your halogen neon laser. That's it. Simple as that. Laser Doc Von, mag-i-emit ng light. Simple as that. Tapos, dadaan yung cells, matatamaan. And then, meron tayong tinatawag na detectors. Ano yung detectors, Doc Von? The purpose of your detectors is to capture the light and to convert it into an electrical signal. So, that's it, no? That's the detectors. Meron na tayong tinatawag na lens blocker. Ang mga lens blocker are provided to prevent non-scattered light from entering the detectors. Kasi usually, ang gusto nating ma-detect sa detector ay yung scattered na light and not the non-scattered na lights. Again, that's simple. And then, we have the different positioning of your light, optical light. Meron tayong forward. Pag forward, nasa harap lang yan siya. Meron tayong side na nasa tabi, or a combination of your low angle scatter or forward high angle scatter, or tinatawag natin na differential na scatter. Sige. Punta muna tayo sa principle. At least na-organize natin yung thought natin. Sige, una-una, as the cell pass through the sensing zone, so pag dumaan na yung cells natin dito sa sensing zone, ito yung sensing zone natin kasi nandito yung laser natin and your detector. As the cell pass through the sensing zone and interrupt the beam. So, of course, di ba may beam dyan? Pag na-interrupt siya or may na-interrupt ng cell natin, what will happen? Light is scattered in all direction. Tingnan nyo, nasa scatter na ang ating light in all direction. Light scatter results from the interaction between the processes of your absorption, diffraction, refraction, and reflection. Again, bakit nagkakaroon tayo ng scattering of light? It's because of the interaction between the different processes. Ano yung processes? No? Light is scattered in different directions because of these different processes. Absorption, of course, we all know, it will just basically absorb. Diffraction, anong ibig sabihin ng diffraction? Bending around corners or the surface of the cell. Refraction, bending because of the change in speed. And reflection, because backward scatter of rays caused by an obstruction. Again, light scatter result from the interaction of different processes. Pag sinabi nating diffraction, meaning bending around the corners of the cell. Refraction, bending because of the change in speed. Reflection, backward scatter of rays caused by backward scatter of rays because of an obstruction. And then, what will happen? The detection of your scattered rays and their conversion into electrical signals is accomplished by your photodetectors. Again, ito na yung photodetectors that will detect your scattered light and convert it into electrical signal. Ito yung tinatawag natin na yung photodiodes, photodiodes and your photomultiplier tubes at a specific angle. So, meron tayo silang mga specific angles. Ano yung mga specific angles nila Doc Von? Meron tayong forward angle scatter, meaning at a particular degree, 0 degree kasi basically straight lang. That's your forward angle scatter. Or your 0 degrees. Remember forward angle light scatter at 0 degrees correlates with the cell volume, primarily because of the diffraction of the light. Again, ulit-ulitin natin. Forward angle light scatter, 0 degrees siya, Doc Von, correlates with your cell volume, primarily because of the diffraction of light. So, ang importante tatandaan, pag we are talking about forward angle scatter, we are talking about the cell volume. What else? We have your side scatter light or aka tinatawag natin na orthogonal light scatter. Your orthogonal light scatter is at 90 degrees. Remember, 90 degrees or tinatawag natin na side scatter, which results from the refraction and reflection of light. So, kung ang forward angle natin are diffraction of light, yung side scatter light naman natin is because of the refraction and reflection of light from larger structures inside the cell. So, since we are talking about inside the cell, therefore, it will correlate now to the internal complexity. So, we are talking about the internal structures of the cell. So, again, kasi nga, yung forward angle, cell volume, yung side scatter, internal structures of the cell. How about your forward low angle scatter or forward high angle scatter? Yung forward low angle scatter at 2 to 3 degrees and your forward high angle scatter 5 to 15 degrees. I'm sorry, sige, ulitin natin. Your forward low angle scatter 2 to 3 degrees and your forward high angle scatter at 5 to 15 degrees will also correlate with cell volume and refractive index or with internal complexity. Meron tayong tinatawag na differential scatter. It is a combination now of these low angle and high angle forward light scatter and is primarily used for cellular analysis. So again, pag cellular analysis, we are talking about your differential scatter. So, important na concept dito sa optical light scatter is to understand that the positioning of your different detectors kasi this will tell us the different property of the cell being detected. Pag dumaan yung cell dito, magdi-diffract yung light or magre-refract or mas-scatter ang light specifically. And because of the detection by the different angle scatter or the different scatter light or positioning of your detectors, we will be able to identify the different properties of cell, talking about the cell volume, talking about the internal structures of the cell. That is the basic concept of your optical light scatter. I hope you were able to understand that concept. So, ito naman ng different variables measured by your optical light scatter. Meron tayong tinatawag ni RBC count, your mean cell volume, your 5-part WBC differential. So, we have another concept that you have to understand. We have your fluorescent flow cytometry. Actually, no, we are already magaling na tayo dito because this is just a combination of your light scattering that we discussed a while ago plus in conjunction with your fluorescence because it will measure multiple cellular and fluorescent properties of cell. Kaya nag-a-add tayo ng fluorescence because we want to detect because particular cells will have its particular fluorescent properties when they flow as a single cell suspension through your laser beam. So, the same principle that we discussed a while ago. So, dadaan yung flow cell natin, yung cell natin dito pa isa-isa. May laser beam tayo and the different detectors that will identify the properties of your cell. Kasi nakalagay yung side scatter light for your internal cell structure, side fluorescent light. So nagdagdag tayo ng another property, guys. So we are using your fluorescence detectors. Kasi yung fluorescence detector will tell us the RNA and DNA information. And lastly, your forward scatter light. So basically, your fluorescent flow cytometry is a combination of your optical light scattering plus your fluorescence. Kasi sa optical light scattering, dinidetect lang natin kanina yung internal cell structure plus your cell volume. But with the addition of your fluorescence, we were able to have an idea about your RNA or DNA. So these are the basic components of your flow cytometry, no? We have your fluidics. So ito, this part, your fluidic system. I know you're already familiar with this one, kung saan dadaan yung ating cells. And then, we have your optical system. And then, we have your electronic system. So first, let's talk about your fluidic system. Yung fluidic system natin, the sample is actually injected into stream of sheet fluid within the flow chamber. Of course, dito dadaan yung ating cells natin. And then, cells are hydrodynamically focused. If we are talking about a high flow rate, that is used for immunophenotyping analysis. Once it is a low flow rate, that is basically for your DNA analysis. For our optical parts naman, of course, nandyan yung we are talking about your laser light that is required to excite your cells. Yan yung another mga diniscuss natin kanina, yung mga light scattering and then the addition of your fluorescence because of a light emission. Again, mga photodetectors na ating pinag-uusapan kanina, which is a system of optical mirrors and filters, then direct the specific wavelength of light to the designated photodetectors. And lastly, we have your electronic system. Yung electronic system naman will convert your optical signals, your photons, to a corresponding electronic signals or your electronics. Your electronic signal produced is proportional to the amount of light striking a cell. Again, yung electronic signal natin na produce is proportional to the amount of light striking a cell. So, ano nangyayari? Paano nangyayari yan? Paano nako-convert from a photon to electronics? Your light is converted to electrical signal. then amplifier receives the electronic current and converts that to a voltage pulse. Again, ano ang sinabi natin? Isang voltage pulse will be equivalent to 1 cell. Your analog to digital converter, or your ADC, assigns the voltage pulse is assigned to a digital value representing a channel. And then the channel number is transferred to your computer, and dyan na yun na di-display ang ating data. So the computer displays now the channel number to the appropriate position to in the data plot. So dyan na natin makikita in ang distribution and characteristics ng ating cells by means of a plot. Pwedeng histogram, pwedeng scatter plot. Sige lang, i-discuss natin yung scatter plot also later. So in fluorescent flow cytometry, these are the important concepts that we have to identify: your data analysis, your gating, and your sorting. Pag sinabi natin data analysis, basically, collection and being stored in the computer using now your forward scatter, side scatter, and your emitted fluorescence. Because remember, these different things that we discuss, forward scatter, side scatter, emitted fluorescence, they will have a specific na mga data or properties of the cell that will be able to collect. And then, of course, if we collect that, we have to analyze those important data. Then, the data plots, if single parameter, that's your histogram. Pag 2 parameter, that's your data plot. Pag sinabi natin gating, this is actually a boundary that can be set to restrict the analysis to a specific population within the sample. The data selected by the gate is then displayed in your subsequent plotting. So again, ipiniplot naman natin yung data natin. And lastly, your sorting consists of collecting cells of interest. So parang kumbaga, sinusort lang natin, defined through a criteria of size and fluorescence. Remember, specific cells will have a specific property, specific sizes, and then meron din silang specific fluorescent na activity. So kumbaga, sinusort natin siya based sa size and fluorescent activities. Kumbaga, sinusort lang natin sila, or sineseparate, or kina-cluster. That's also the appropriate clustering or sorting of your cells. Again, one important property or usefulness of your fluorescence is for the estimation of your reticulocyte count. It's actually based upon the various dyes and fluorochromes by your RNA. So this is now the estimation of your reticulocyte count. So these are the various dyes that we use. We have your auramine. I know you are familiar with this in your histopathology. So Doc, ano palang purpose ng reticulocyte count? So again, the purpose of your reticulocyte count, it will provide us a quantitative measure of your erythropoiesis. Remember, if you have a certain degree of anemia, then therefore your body will react on that kind of scenario. So therefore, you have to create more RBC through erythropoiesis. And since your reticulocyte counts will provide a quantitative measure of erythropoiesis, remember, your reticulocyte count is a younger red blood cell. So therefore, the more erythropoiesis, because we need more RBC, the younger the cells that are also being produced. So mas marami tayong nakikitang reticulocyte count. Therefore, it will now give us a quantitative measure of your erythropoiesis. Again, anong concept na dapat nyo maintindihan in terms of your estimation of your reticulocyte count? The degree of your fluorescence emission caused by the excitation bound dye is proportional to the amount of RNA in your erythrocyte. Again, the degree of your fluorescence emission caused by excitation bound dye is proportional to the amount of your RNA in the erythrocyte. So again, the reticulocyte fall into, pwede siyang low fluorescent, middle fluorescence, or high fluorescence reticulocyte. With less mature reticulocyte, pag less mature siya, it will show higher fluorescence. Therefore, more uptake of your RNA in the erythrocyte. Again, the degree of your fluorescence emission caused by the excitation of your bound dye is proportional to the amount of your RNA in your erythrocyte. Therefore, with less mature reticulocyte, or the more immature ang ating reticulocyte, it will show your higher fluorescence. So, again, yung higher fluorescence, the more less mature or the more immature ang ating reticulocyte. So, remember that concept. So, what are the other methods? Meron tayong peroxidases method, fluorescence method, and your immunological method for testing. Yung peroxidase, basically, identification lang and counting of your granulocyte. Your lymphocyte are not stained. We have also your fluorescence, your reticulocyte, and platelets, best for detecting your immature platelets. And we have your immunological testing for your accurate platelet counting using the different CD4 antibodies, your CD41 and CD61. This is actually the summary, no, of the things that we discussed a while ago. So, ito yung sinasabi na. So, lakihan ko lang ang diagram ha, para ma-appreciate nyo. Yan. Lakihan ko pa para ma-appreciate. Ayan, malaki na. These are actually the different principle na ginagamit ng mga different machines. So, ito yung Beckman and Coulter, your Sysmex, your Abbott, and your Siemens. Ito yung different na mga machines or brand manufacturers ng ating mga analyzers. Ang pinaka-common na naka-encounter, of course, na-encounter ko nitong apat na to na different hematological analyzers. Pero pinakasikat talaga is yung Beckman and Coulter kasi sila talaga yung nag-umpisa sa lahat. So, dapat nakalagay dito ang mga different parameters na detect ng ating different manufacturers, no? Yung WBC, yung RBC, yung hemoglobin, hematocrit. So, nakalagay din dito kung anong mga principle na ginamit nila to detect that test. For example, your RBC counting. For Beckman and Coulter, gumagamit sila ng impedance. Yung impedance. Pero sa Siemens, ang ginagamit nila, your low angle and high angle laser light scatter. So for example, yung hemoglobin, ginagamit is your modified cyanamethimoglobin, your modified cyanamethimoglobin, or iba, sodium lauryl sulfate. Again, don't have to memorize this one. No? Hindi ko nga to memorize eh. And I guess, hindi naman kailangan na you have to really memorize this one. Ang purpose lang ng table na ito is to show you the different principle that we discussed a while ago. Pag Beckman and Coulter, ang ginagamit sa WBC is your impedance volume. Pag RBC, yung impedance pa rin. Yung hematocrit, yung formula na to. Yung platelet, still your impedance. So, yung reticulocyte count, talking about your supravital staining. Pero sa Sysmex, ang ginagamit nila, yung diniscuss natin, yung fluorescent staining, etc. Again, these are just the different principle that is being used by the different manufacturer of your automated hematological analyzers. Now, let's talk about histogram. So, ito yung sinasabi natin, di ba? Yung histogram natin kanina, ito yung nakikita natin ay graphical representation of your numerical data of your different cell populations in a cell counter. So, ano nakukuha natin sa ating histogram? Nakakuha tayo ng data in regards to the average size of the cell as well as the distribution of the size. So, ito yung mga importante. So, pwede tayong makakita. So, again, ang x-axis that talks about the volume of cell. So, kaya ang nakalagay dito 30, 200. This is, ang unit nito is your femtoliter. And then, your y-axis that refers to the number of cells. Kumbaga, pag mas mataas ang peak, so mas mataas. So, ang RBC in terms of the number, mas marami compared sa platelet. In terms of the volume, mas malaki ang volume ng ating RBC compared sa ating platelet. So, that's how you interpret, basic interpretation of your histogram. Now we have your discriminator. So yung discriminator natin, magsaseparate sa ating distribution curve for the volume. For us to differentiate whether that is WBC, RBC, or your platelet. So again, what is the purpose of your discriminator? Will tell us or will separate the distribution curve for that particular volume. Kung WBC ba yan, RBC ba yan, and your platelet discriminator. So pag sinabi nating WBC discriminator, may dalawa tayong tatandaan, yung lower discriminator and your upper discriminator. Yung lower discriminator for your WBC, ang range is around 30 to 60 femto liter. Pag upper discriminator naman for WBC, that is fixed at around 300 femto liter. Pag sinabi nating RBC discriminator, the lower discriminator is around 25 to 75 femtoliter. The upper discriminator is around 200 to 250 femtoliter. And lastly, for your platelet discriminator, meron tayong tinatawag na, again, your lower discriminator of 2 to 6 femtoliter and upper discriminator 12 to 30 femtoliter. And a fixed discriminator of your 12 liters. Pag sinabi natin fixed, but if it's 12 femtoliter, that's automatically your platelet. So, kumbaga, for example, ito. Again, let's review nga. Sabi natin kanina, sige, compare natin yung platelet. I-compare natin yung ating platelet sa ating RBC. So, sinabi natin sa RBC natin, ang lower discriminator natin is around 25 to 75. So, RBC, so pasok, no? 25 to 75 femtoliter. Ang upper discriminator naman natin sa ating RBC is 200 to 250. So, within this range, so somehow, pasok pa rin siya sa around 200 to 250 na parameter. And lastly, for fix sa ating platelet, lower discriminator, 2 to 6 femtoliter. So, pasok. Lower discriminator is around 2 to 6. And your upper discriminator of 12 to 30. So, this is your pasok pa rin siya sa ating upper discriminator. And the fixed platelet discriminator of your 12 femtoliter. So, again, a platelet size distribution plot is produced using a 3 threshold. So, ito yung 3 threshold natin tatandaan. One fixed at the 12 femtoliter and the other 2 are allowed to hunt the upper and lower ends of your platelet population between certain limits. The lower platelet size threshold may move between 2 and 6 femtoliter, the higher between 12 and 30 femtoliter. So, the purpose of Doc Von, ano bang importance ng ating mga threshold or mga discriminator? Again, the purpose of these different threshold na we discuss about is to distinguish your platelets from the small red blood cells that is located in your upper end of your platelet population and debris at the lower end. Kasi nga, remember, magmi-meet sila dito from the lower discriminator of your RBC and the upper discriminator of your platelet. Dito sila mag-meet. So that's why it is important for us to have these threshold or discriminators para ma-distinguish natin ang kanilang mga particular boundaries. In this example, the purpose of this threshold to distinguish now your platelets from your small red blood cells. Kasi dito, mas maliliit yung RBC sa bandang area na ito. Sa lower limit ng ating RBC, lower discriminator ng ating RBC. Meron din tayong tinatawag na shift to the left in terms of your RBC histogram. Very basic lang, no? Pag shift to the left, a left shift curve, this is the normal curve of your RBC histogram. Pag shift to the left, it means there is a presence of microcytosis. Kasi nga, kung normal ito, mas maliit kung nandito ng ating cells, nasa left side going to 0, meaning mas maliliit ang mga cells, right? Kasi value dito, 0 to 50. So therefore, RBC is smaller. So therefore, it is microcytosis. I guess nagme-make sense naman siya, no? Kasi ang value dito, 0, 50, 100, 200. Pag shift to the right, meaning more than the normal distribution, therefore mas malalaki ang ating RBC, kaya tinatawag natin na microcytosis. And then we have also a dimorphic population. Pag sinabi nating dimorphic population, there is a bimodal peak of the curve. So two population of sizes of your red blood cells. So tingnan natin, may isang peak, may another peak. This is the bimodal peak of the curve. And also, we have now your WBC histogram. Kanina, we discussed about your platelet, your RBC. Ito naman, this is now your WBC histogram. Sa ating WBC histogram, take note that cells that is greater than 35 femtoliter are counted as WBCs in your WBC and hemoglobin chamber. Your cells with volume, pag ang volume ng ating cells is around 35 to 90 femtoliter, that is your lymphocytes. Pero ang cells natin with 90 to 160, that is your mid cells. Kasi mid cells in between your lymphocyte and granulocyte. And these mid cells refer now to your monocyte, basophil, and your eosinophil. So remember this value, 90 to 160 femtoliter. And lastly, we have your neutrophils. Your neutrophils naman natin is around 160 to 450 femtoliter. So for example, so again, cell volume 35 to 90. So tingnan natin, 35 to 90. This refers now to your lymphocytes. 90 to 160, that refers to your mid cells or between cells. That's your monocyte, basophil, and eosinophil. And then lastly, for your neutrophils, 160 to 450 femtoliters. That's your neutrophils. This is actually the normal WBC distribution curve. However, we also have an abnormal WBC histogram. Pag mamatataas yung pag may nagpa-particular na nagpipeak lamang, mabababa yung ibang value, tapos nagpipeak yung particular parameter, then we will be able to identify what type of lymphocytosis. So this diagram shows the different types of your leukocytosis. Pag mataas ang neutrophil, that's your neutrophilia. Pag mataas ang lymphocyte, lymphocytosis, monocytosis, eosinophilia, and etc. However, sige based sa tatlong parameters na to, sino ang mahirap i-detect? This parameter, your lymphocytes, this one, your mid, or your neutrophils. Ano kaya dyan? Alin kaya doon ang mahirap i-detect? Your mid cells, your neutrophilia, or your lymphocytosis? Sino ang mahirap ma-distinguish? Again, the answer is yung between cells or yung mid cells. Bakit? It is difficult to distinguish this area if there is a presence of lymphocytosis. Kumbaga, may monocytosis, may increased basophils, may eosinophilia, because remember, the W distribution of that particular curve are all distributed in the same region. So therefore, pag nag-peak dito, mababa yung lymphocyte, mababa yung neutrophils, tapos dito bumigla nag-peak, madidetermine ba natin whether monocyte yun, basophil, eosinophil, or a combination of cells? Of course, the problem is that we cannot able to distinguish kung ano kaya ang nag-increase doon. Kasi nga, yung monocyte, basophil, and eosinophil is located in the same area. That is just the basic concept kung sino ang mahirap ma-distinguish. Kasi nga, for example, if you're talking about neutrophilia, alam naman natin yung neutrophilia is around 160 to 450. So therefore, pag may nag-peak around 160 to 140, mabababa yung other parameter, that is neutrophilia. If lymphocytosis naman, your lymphocytes, is around 35 to 90 femtoliter. 35 to 90 femtoliter. So, nag-peak siya dito, mababa yung other parameters. So, therefore, we all know that this is lymphocytosis. However, pag nasakit na ang problema, yung mid cells, we will not be able to identify kung ano ba talaga. Whether it is monocytosis, eosinophilia, or your basophilia. But we all know that these three cells could be the reason of the increase of that particular parameter. I hope you understand that concept, no? So, again, so let's have a exercise. So, identify the type of leukocytosis of your IWBC histogram. So, can you interpret this one? Is this neutrophilia, basophilia, monocytosis, eosinophilia, or lymphocytosis? Of course, again, check muna kung anong volume natin. Oh, nasa 50, 35, tapos nasa 90. So, this range is your lymphocyte. So, tapos, mabababa yung other parameters. Nag-peak lang siya. So, therefore, this is lymphocytosis. Very good. How about this diagram? This diagram, Doc Von, 160 to 450. So, around 160 to 300. Napasok siya sa neutrophilia. Neutrophilia siya kasi nag-peak siya, tapos yung other parameter is abnormal. So, but please remember what is the normal curve. Ito yung normal distribution curve. Pag biglang nag-peak lang dito, tapos mababa yung other parameter, that is already lymphocytosis. Pag mababa dito lahat, pero mataas ang neutrophils, therefore, that is neutrophilia. But ang problem natin is yung mid cells, no? Kung dito yung tataas kasi we're not able to identify kung ano kaya sa tatlo ang nag-increase. Pwede bang combination? Yes, pwede. Pwede yung tatlo talaga nag-increase. Pwede, or pwede lang mang na isang parameter. So that is your WBC histogram. Now, let's talk about your WBC scatter plot. So, usually, no, makikita nyo to sa mga laboratory result, no, ng mga, if you are using mga Abbott, Sysmex, ganito ang nakikita natin. May mga diagrams. So, tingnan nyo nga yung mga WBC results nyo, yung mga high-end na mga machines, merong mga ganitong diagrams, including your histogram. Actually, nakikita yan sa mga other laboratory na mga results, no? At least you already know how to interpret those histogram. Pero usually, no, ang doctors hindi naman tinitingnan talaga sa mga diagrams kasi we just look directly to the result kasi mai-interpret by just knowing the normal range. Lymphocytosis to, mataas siya. Pero, it's just nice for us no na we all know how to properly interpret your histogram. So, meron din tayong tinatawag na scatter plot. So, paano ba natin na-interpret tong scatter plot na ito? So, these are the different scatter plots. So, just take a look at this diagram, your scatter plot. Yung scatter plot natin, meron tayong tinatawag na yung x-axis refers to the side scatter. Yung side scatter will detect the granularity of the cell. Sa ating y-axis, meron tayong forward scatter that will reflect the size of the cell. So, again, x-axis, side scatter, granularity. The more padulong siya sa right, meaning more granular. Pag sa ating y-axis, that refers to your forward scatter. So, pas-akyat ang ating diagram, meaning the more malaki ang kanyang size or the larger the size. So, let's try to interpret this diagram. Anong nakikita natin? Your lymphocytes, yung red na to, this is actually your lymphocytes. Your lymphocytes are smaller and have few internal components or granules than your monocyte and your granulocyte, which is the purple. Again, mapapansin natin na yung lymphocyte dito, they have a smaller and have fewer internal components kasi nga nandito siya sa side na to, so mas mababa ang granularity kasi i-increase mo to, yung more granular siya, pero nandito lang siya na part, so therefore lesser granularity and mas maliit siya compared sa ating monocyte and your granulocyte. So again, your lymphocyte are smaller and have fewer internal components or granules than your monocyte and your granulocytes. Your granulocytes naman, anong masasabi natin sa ating granulocytes? Most granular population kasi nandito na siya na part. So therefore, your granulocytes are the most granular population and they have the most side scatter and appear farthest to the right on the scatter plot. That is how you simply interpret your WBC scatter plot. You just take note kung ano yung mga properties na nakalagay sa ating diagram. Sa x-axis, side scatter granularity. Y-axis, forward scatter, the sizes of the cell. That is how simply interpreting your WBC scatter plot. Usually kasi may mga iba sigurong doctors na they don't prefer this data na gusto nila makita talaga yung diagram like this. Pero ako personally, automatically dito ako sa data. Okay, like ako namang interpret 6.8. That's normal. I don't want to, kasi hindi ako marunong dati. Hindi ako, technically speaking, no, because of this lecture, doon ko pa naaaral yung mga RBC, histogram, and your granularity. I'm also learning. So, learning is fun. Ironic, no? Doctor na ako, gumaduate na ako ng medtech, doctor na ako, pero ngayon ko pa lang siya naintindihan yung mga different na mga histogram, yung mga scatter plan. Usually, nakikita ko lang talaga yung medtech ako dati, nag-duty ako. Nakikita ko lang to, and hindi ko siya na-interpret. Kasi ako, technically, dito lang talaga ako sa data tumitingin ng result. Pero no, that's the importance, no, it's the visual. Kumbaga, the importance of having this scatter plot in your histogram is a visual representation of the result. So this is now the data and you want to see that as a visual representation using your histogram or your scatter plot. Ayan, dito, ganito. Ito yung mga different laboratory na mga na nire-release natin. Meron tayong makakita na scatter plot, and your mga histograms. So at least now you're able to understand how to interpret this one. Just know the different important parameters kung ano yung nakalagay sa x-axis and your y-axis, no? Marunong na tayo dito. Very good. Now, let's talk about your calibration. Of course, we have to calibrate our machine, no? Your QC/QE control. Of course, every morning before we run our test for that particular day, we have to do your calibration because your calibration will ensure readings from your instrument that are consistent with other measurement. Will also determine the accuracy of the instrument readings and to establish reliability of your instrument. So this will determine the accuracy and precision of the analyzers. Somehow this tinatawag natin na tuning up of your instrument. So this is done upon installation of your machine and in every 6 months. So pag calibration every 6 months lang yan, pero yung mga QC, QA, that's every day. And there is a periodic after certain major repair. Again, we're down to our last discussion. We will be talking about the limitations and interferences. So these are the different factors that will affect your hematology analyzers. Kung baga, ano yung mangkukos ng mga erroneous results. So isasayin natin, lakayin natin ng diagram natin. Of course, now, these are the conditions that will cause interferences on most of your hematological analyzers. So another conditions. First condition, we have your cold agglutinin. So anong may effect pag meron tayong cold agglutinins? Of course, it is an agglutinin. Therefore, it will cause agglutination of your red blood cells. So mag-agglutinate, magkukumpul-kumpulan ang ating mga RBC. Therefore, magkakaroon ng pag magkukumpul-kumpul ang ating RBC, anong mangyayari? Bababa ang result ng ating RBC, tataas ang MCV volume. Bakit? Bakit tataas ang MCV volume? Kasi nga, nagkukumpul-kumpulan, nagmumukhang malaki ang RBC. Kaya, ang mean cell volume parameter will also be increase, including your MCHC, kasi parang nakakount as 1 yung cell, and it will create your grainy appearance. So, how can we correct this problem? We just simply warm the specimen. Since it's a cold agglutinins, you just have to warm your specimen at 37 degrees Celsius, and you rerun the test. So what is the instrument indicators na nakikita natin? Of course, nakikita natin there is a dual RBC population on your RBC map, or right shift on your RBC histogram. Pag right shift gana, ano yung right shift? There's already macrocytosis, kasi nagkukumpul-kumpulan ng cell, nagmumukhang malalaki ang ating cells. Kaya macrocytosis. Dual RBC population, anong tawag, anong nakikita natin sa ating histogram dito? There is a dimorphic population, or therefore there is two presence of different sizes of your RBC. Dalawang population or sizes ng ating double RBC. Next parameter, lipemia or your icterus. Anong nangyayari sa ating lipemic? Masyadong lipemia, masyadong mataba. Therefore, there is increased turbidity that will affect your spectrophotometric reading for hemoglobin. Since your spectrophotometric principle will be related to the light being absorbed. Pag most turbid, pag mas turbid siya, therefore, more light ang ma-absorb. Therefore, mas may increase ang absorbance, mas may increase ang result. Therefore, ang result will be increase hemoglobin, increase MCH. Instrument indicator natin dyan, of course, hindi na siya nagtutugma sa ating rule of 3. So, how can we correct such problem? Simply, you just replace the plasma. How about hemolysis? Of course, once our cells are being hemolyzed, ano nga nangyayari sa ating RBC? Nasisira ang ating RBC. Therefore, ang parameters na affected, obviously, mababa ang RBC, mababa ang ating hematocrit. And also, it will have a problem now sa ating rule of 3. And all you have to do is request new specimen. Kasi nga, hemolyzed na, no? Mali na ang ating RBC counting and your hematocrit counting. Next, how about your lysis-resistant RBC with abnormal hemoglobins? So, rationale, your RBC with hemoglobin S and C or F may fail to lyse. Of course, hindi nalalyse yung ating yung mga RBC na may ganitong hemoglobin problem. And it will be counted as WBC. So, again, yung abnormal na mga hemoglobin ng ating RBC, hindi nalalyse ng ating specific reagent para makount yung WBC. Since hindi siya malyse, therefore, nababasa siya sa ating machine as WBC. Kaya tumataas ang ating WBC. Of course, your hemoglobin parameter will also be affected, causing an increase of your hemoglobin. So, how do you correct such problem? You perform your manual dilution or allow incubation time for the lysis. Another parameter, your microcytes or schistocytes. Masyadong maliliit, di ba na? Microcytes, maliliit na cells, or your schistocytes, maliliit na cells. So, volume of your RBC or your RBC fragments less than the lower RBC threshold and or within the platelet threshold. So, kumbaga, masyadong maliliit ang RBC. Nababasa siya as platelet. Kasi, we all know your platelets are maliliit. So, yung RBC na maliliit ng microcyte and schistocytes, nababasa siya as platelet. Therefore, your platelet will be increased and your RBC will be decreased. Kasi yung maliliit na RBC, hindi siya nababasa as RBC, kaya bumababa ang RBC. Pero yung maliliit na RBC, na me-measure siya as platelet, therefore, tumataas ang ating platelet. So, how can we correct such problem, Doc Von? Simply, you make a blood smear and review that in your microscope. So, manual checking again. Again, these are the last parameters that we have to take note. Your nucleated RBC, your megakaryocyte fragments or your micromegakaryoblast, your nucleated RBC or your micromegablast counted as WBC. So, itong mga parameters na to, naka-count siya as WBC. So, how can we address such problem? Newer instrument, eliminate these errors. So, mga high-end na mga instrument na i-eliminate na ito ng problem na to. However, count the nucleated RBC and correct your WBC count. So, basically, you will just do manual counting. How about your platelet clumps? Your platelet clumps will cause a large clumps counted as WBC, not platelet. So, nagkukumpul-kumpulan ng ating platelet, nababasa siya as WBC instead of platelet. Therefore, bumababa ang platelet, tumataas ang WBC count. So, what do we need to do? You redraw the specimen in your sodium citrate and multiply your result by 1.1. So again, kukuha ng bagong specimen in a sodium citrate. Anong tubes ito? Red, black, green, or your blue tube. And you just multiply the result by 1.1. Pero kung ako hindi na lang ako mag-redraw. I'll just do manual counting. Mas madali yun. Next, if WBC count is greater than 100, so masyadong mataas yung WBC count natin, it will cause a turbidity that will also affect your spectrophotometric reading for your hemoglobin, WBC count, and RBC count. Therefore, hemoglobin will be increased, RBC will be increased, and there is an incorrect result of your hematocrit. So, parameter, so paano natin siya mako-correct? Simply, you manual checking of your hematocrit. So, you just do manual checking of this parameter. Next, leukemia, especially with chemotherapy. So nag-undergo ka ng chemotherapy. So, therefore, your cells, your WBC are very fragile. So, it will be fragmented and counted as platelets. So, bumababa yung WBC count, tumataas yung platelet. So, again, all you have to do is review your film or manual check. That's why, kahit sa mga ganitong mga parameters sa ating patient, we have to really know the diagnosis of our patient. Kasi usually nga sa mga requests sa mga inpatient, meron nakalagay na diagnosis and you will be guided with your results. That's why if there's something abnormal or not correct with the CBC results na print ng ating machine, you have to really investigate. Hindi lang pwede na baka may error ang machine, baka may Q's and Q. What if the problem is in the patient? Because there might be a underlying causes sa ating patient na that will cause the certain result. So that's why you have to investigate. Check again your the diagnosis of the patient, because the data will give us a hint of the possible problem. And the last parameter is the old specimen. So once it's an old specimen, your RBC will swell as a specimen ages, your platelets swell and degenerate. Your WBC is affected by the prolonged exposure of your EDTA. So what will happen? Mataas yung MCV, MPV, bumababa yung platelet, and automated differential may be incorrect. So how can we do that? Simply establish stability, and of course we have to reject the specimen or get a new specimen. Yes, that's the end of our discussion for today. I hope that you were able to appreciate the importance of this automation sa ating hematology. Again, if you have any questions in terms about our topic, please do not hesitate to post your questions in our content forum. So, guys, good luck with your quiz next week. Thank you for listening. God bless everyone and happy Aral.